Hello everybody. Today we will start with the new chapter in biology for standard 9th ICSE, that is economic importance of bacteria and fungi. It's a 9th chapter in a case of textbook Concise biology. Now this year we do not have the chapter 5 kingdom classification. It has been cancelled from your syllabus, but in the 5 kingdom classification we come to know that the classification of the living organisms was done into 5 categories. Okay, so in those categories which are there, out of those 5, 2 are the one which are bacteria and fungi. So that's why we have this particular thing that plants and animals are always there, but apart from the plants and animals, there are another 3 kind of classifications in case of the human the living organisms, and these classifications are given as the as the Monera, Protista and fungi. So Monera, Protista and fungi are the ones. So out of this Monera and Protista that will be coming under category of the bacteria and all, and fungi is of course a complete category by itself. So we see that the bacteria and fungi are being given a separate kingdom, or you can say we got 5 kingdom. They are given separate kingdom because they could not be said to be as an animal, nor they could be said to be as a plant. So they were certain kind of organisms which are completely different from plants and animals, and that's why it was necessary to list them as a separate kind of a kingdom altogether, and that's what we are going to that's what we would have been studied in the 5 classification, but since it is not in the portion, so we'll just go for the bacteria and fungi as a separate chapter altogether. So bacteria are primitive organisms made up of single cells. So yeah, the chapter is divided into 2 parts. One part will be completely about the bacteria and the second part will be on fungi. So first of all, what we will be having is the economic importance of bacteria. Now bacteria are the most primitive kind of the organisms because they are the one which are single celled. Okay, they are the one which are single celled, and that's also their genetic material is dispersed in the central region of the cytoplasm without a nuclear membrane. That is, they are prokaryotic that lack a true nucleus. So that's what we see is that bacteria are prokaryotic kind of cells. So prokaryotic kind of cells are the one which are primitive in kind. That means they do not have a proper nucleus. Okay. Now the eukaryotic has got a proper nucleus of its own, and that's the genetic material, the DNA and all are present inside the nucleus. But in case of the bacteria, they are prokaryotic. In prokaryotic kind of cells, the genetic material is a part and parcel of the cytoplasm. So that's why they do not have a distinct kind of a nucleus in them. So according to the 5 kingdom classification, bacteria are not plants. The reason being, bacteria are prokaryotic, single cell organism instead of being eukaryotic and multicellular. So that's the reason why they are not placed in the plants card, because plants are eukaryotic and plus plants are multicellular, whereas bacteria are prokaryotic and they are single cell. So let's see the bacteria, a general study. Bacteria are the most primitive unicellular organism. Each bacterial cell contains a single chromosome nuclear material which is not enclosed in a nuclear membrane. So the first and foremost thing is that it has a single cell. Secondly, it does not have a proper nucleus. It has got the genetic material inside the cytoplasm and which is not enclosed in a nuclear membrane. Bacteria are so numerous and everywhere. Now that's a fact. Bacteria is present each and every place. It is just everywhere, inside our body, outside on our hands, in the air, everywhere. Okay, so bacteria are present almost everywhere. There are several thousand known species of bacteria which occur in air, water, soil, food, on or inside the body of a living organism, and in short, practically everywhere. In our body, there are hundreds and thousands of bacteria in the mouth, on skin, inside intestine, inside in the genital tracts, etc. some bacteria occurring in our intestine are beneficial. They synthesize certain vitamins such as vitamin B. Certain bacteria are harmful and cause many diseases, but there are others which are highly useful as well. So when we talk about bacteria, yes, they are present everywhere in our body. So they are present on the skin, they are present inside our mouth, they are present inside the respiratory tract, it is present inside the intestine. So everywhere their presence is going to be there, but it is not necessary that the bacteria will only be harmful. There are bacteria which are good for our body and they are helping us to synthesize the vitamins from the food what we intake in. So that, for example, vitamin B is being synthesized by the bacteria in the intestine. At the same time, there are bacteria which can be really harmful for our body. So there could be both kinds of bacteria, harmful as well as useful bacteria, which could be either harmful for the body or useful for the body. A simple example would be curd. Curd is good bacteria which are going to help us in digestion. According to one estimate, there are more bacteria normally living on our skin than the total number of humans living on earth. So the population of the that's a main, it's quite a remarkable thing that the total population of the humans on the earth, okay, usse zyada is a number of bacteria present in our body or our body. So on our skin, so that's a huge amount of bacteria. These bacteria are even useful as they prevent the growth of harmful ones. So bacteria, many things are going to be they are going to be useful because some of the bacteria do not allow the other bad bacteria to grow over there. So that's the reason they sometimes become more beneficial for the body. Talking about the size, the bacteria are smallest living organisms on earth with an average size of 2 micrometers long and 0.5 micrometers thick. 1 micrometer is 1,000th of a millimeter. So you can imagine 1,000th of a millimeter, and it is just 0.5 micron thick and 2 micron long. So that's why you can see that they are absolutely very small organisms. Shape-wise, the bacteria are usually of 4 types. Cocci are spherical bacteria. So if they are round circles like this, then there are cocci. Okay, that is spherical. Bacillus are rod-like. So if they are something like this, then they are bacillus. So this is spherical. This is rod shaped. Okay. The next is spiralia. So spiralia will be spiral like this. So this is spirilla, spiral. Okay, and or twisted. And the last one is comma shaped. So it's like this. So this is comma shaped, which is vibrio. Okay, which is comma. So hence we will find that the bacteria can be of these 4 kinds, that is cocci, spherical, bacillus, rod shaped, spirilla, spiral shaped, and comma shaped is vibrio. Now, not only that, they are also going to live in a different form. So some of them will be in clusters, some will be in a chain. So depending upon how they are going to be living together, they will be identified in that manner. So first is, many bacteria live singly, but some occurs in pairs. That is why it is diplococci. So if it is these 2 are in pairs, so because they are in pairs, that's why it is diplococci. Okay, or in a long chain. So if it is in a long chain, so if it is like 1, 2, 3, 4 as a chain long chain connected to each other, that will be streptococci. So this streptococci. Or in cluster will be staphylococci. So if it is in a cluster, so agar aise clusters mein hai, so it will be staphylococci. So in this way, if it is of course cocci is for the spherical one. If they would have been rod shaped, then it will be Diplobacillus, Streptobacillus, Staphylobacillus. So depending upon how they are going to be placed in bunches or it will be placed that way. So that's why it will be of that form. So many bacteria are single, some occur in pairs, diplococci, or in long chain, streptococci, or in clusters, staphylococci. So they are given the examples over there. And then is the structure. Each individual bacterium is rather a simple cell. So yes, the first thing is that they are very, very simple cells. Okay. It contains the living substance, protoplasm or protoplasm lying inside the cell membrane, cytoplasm plus the bacterial chromosomes. Now see over here, we do not call it as the cytoplasm because cytoplasm would not include the nucleus. Okay, we have understood in the cell chapter that the cell ke andar, the contents of the cell are divided into two parts. One is the cytoplasm, which is not containing the nucleus, and the nucleus containing the DNA material. And we also studied that nucleus के साथ में जो content है, that is called as protoplasm. So here we see that the cell is having everything combined in a same unit. So the nuclear material, or we can say the DNA structure, genetic material, is a part and parcel with that particular other kind of organelles which are there. So hence it is the protoplasm and not the cytoplasm. So the cytoplasm plus the bacterial genetical material combined together gives you the protoplasm. So protoplasm or the protoplast lying within the cell membrane, that is cytoplasm plus bacterial chromosome, surrounded by non-living stiff cell wall. So there will be a cell wall and this cell wall is made up of peptidocycline. So it is peptidocyte and not of cellulose as in plants. So the difference between the cell wall made in plants and cell wall made in bacteria is that cell wall in case of plants is made up of cellulose, whereas the one in the bacteria is made up of peptidoglycan. So peptidoglycan are a kind of peptides or proteins by which it is being made. A thin cell membrane lies immediately beneath the cell walls and surrounds the cytoplasm. So there is a thin cell membrane lies beneath. So there is a cell wall. Cell wall के अंदर है cell membrane. So if we see the normal structure of a bacteria, it will be seen that bacteria's normal structure will be something like this, that there will be a cell wall. So this is a cell wall. So that will be a thick cell wall which is made up of the peptidoglycan. So that will be the thick cell wall. Yeah, this is a thick cell wall. And inside, so this is the cell wall. And inside is a very thin membrane which is the cell membrane. So the cell membrane is over here, which is the cell membrane. Okay, and you will find that the chromosomes are within inside lying over here. The chromosome, so this is nothing but the bacterial chromosome which is present over here. And apart from that, so this will be the cytoplasm, and there will be certain kind of granules which will be of glycogen. So glycogen granules, and then there will be certain plastids over here. Plasmids, okay, not plastids, plasmids, so part of plasma, and cell wall and the capsule is there. So this is the cell wall, and also outside will be called as capsule. And then it has got two kind of antenna kind of thing, which is nothing but the flagellum. So flagellum. Flagellum is the one which is helping it to move. So the movement of the flagellum is going to help it to move from the. So of course the cell membrane, the cytoplasm and chromosomes are going to form the protoplasm. So this combines to form the protoplasm, right? And yeah, that is about the structure, bacterial function thing over here. So we just go through it. A thin cell membrane lies immediately beneath the cell wall and surrounds the cytoplasm. Okay, thin membrane, that is the cell membrane. There are no well-defined nucleus, but chromatid DNA material is present in the central region. This chromatid material is in the form of a single circle and is not enclosed in a nuclear membrane, but it is attached to the cell membrane at some point. So this one is attached to the cell membrane at some point. So yes, that would be attached to the cell membrane at some point, maybe something like here. So that will be attached to the cell membrane at some point, but it is not connected, or it is not having a nuclear membrane. So there is no nuclear membrane, and it is just a single circle of the chromosomes. The cytoplasm consists of vacuoles, granules, and in some cases chlorophyll as well. So there could be some vacuoles, there could be the granules, and also chlorophyll in some of the bacteria. Sometimes a slimy protective layer called capsule is present outside the cell wall. So sometimes outside the cell wall is a slimy protective layer called as the capsule. So cell wall ke bahar ek protective layer which will be called as a capsule may also be present in certain kind of bacteria. Movement wise, most bacteria cannot move about on their own, and they are only passively transported by wind, water or contact. So normally the bacteria cannot move by their own. They are going to move with the body which is in contact with, or the movement of the air or the water or the body. But some bacteria possess one or more whip-like flagella which pierce through the cell wall and capsule. The lashing movements of the flagella provide active locomotion in the liquid environment. So in some of the bacteria, they have got these kind of flagella which is coming out from the cell membrane and pushing out from the capsule, and the movement of the flagella is allowing the movement of the bacteria in liquid kind of a medium. So when it's a liquid medium or liquid environment, the bacteria will be able to move around using the flapping movements of the bacteria, of the flagellum. Nutrition wise, most bacteria have no chlorophyll, therefore they depend on ready-made food from different sources, heterotrophic. In this category, the bacteria may be saprotropic, drawing nourishment from decaying dead organisms, or parasitic, drawing nourishment from the body of their living host. So bacteria, most of the bacteria do not have chlorophyll, and that's why they do not prepare their own food. So they will be dependent on some another kind of an organism for their nourishment. So that is why they are not autotrophs, but they are heterotrophs. So heterotrophic in nature, that means they are going to take the nutrition from some another organism. Now when they are dependent on others, they will be of two kinds. One will be a saprophyte or saprotropic. Saprotropic will be the one in which they will be like scavengers, that they will be living on the dead and the decaying substances. So when the bacteria is going to feed itself from the dead and the decay, they will be saprophytic in nature. Whereas if it is parasitic, means if they are going to live on the living organism and take nutrition from that particular organism, then they will be called as parasitic in nature. So the bacteria secretes powerful enzyme from their cells into the surrounding food containing material. The enzyme make the food material soluble, which is then absorbed by solution in the bacterial cell. So for the nutrition to be absorbed into the body of the bacteria, it is seen that bacteria is going to have a external digestive system. What is the external digestive system? That they are not going to intake a material and going to digest it, but they will convert the material, what they are going to eat, what they are going to feed on, they are going to attack on that particular thing, and they are going to break down into simpler substances which can easily be absorbed by the bacteria. So what they do is that bacteria are going to give out some enzymes. So there are some powerful enzymes are going to be given out by the bacteria on the food material which they want to eat, and that would automatically break down that particular complex food material into simpler ones which can be absorbed by the bacteria as nutrients. So that's why they have written over here, the bacteria secrete powerful enzyme from their cells into the surrounding food containing material. So on the surrounding food containing material, it is going to give out the powerful amount of enzymes over there, and this enzyme make the food material soluble, which is then absorbed as a solution into the bacterial cell. So this is how the cells are, the bacteria is going to be dependent on, means the digestion of the food material is going to be taken outside, and it's only the nutritive part which will be taken in by the bacteria, and not the, it's not like this. So they don't need to excrete out, no? Because they are going to eat only the thing which is required by them. The next is about respiration. So some bacteria respire by absorbing atmospheric oxygen, that is aerobic respiration. Other needs no free oxygen, that is anaerobic. The anaerobic respiration are killed if anaerobic bacteria are killed if exposed to air. So the bacteria can be considered can be categorized into two kinds of bacteria depending upon that kind of respiratory system. If they are going to respire in presence of air or in presence of oxygen, so in that case it is atmospheric oxygen वो लेंगे और वो जैसे हम लोग respire करते हैं taking oxygen given out carbon dioxide, that is precisely what they will also do, and that's why they will be aerobic kind of bacteria. Whereas there are certain kind of bacteria which will respire anaerobically, that means in absence of oxygen. So they do not take in oxygen from the atmosphere, and in fact if they are coming in contact with the atmospheric oxygen, they would die. So that's the reason bacteria, these bacteria will be such, they will be always placed in the medium where the oxygen is not available to them directly. So the anaerobic bacteria, that's why they are called as the anaerobic bacteria, which will be respiring only in absence of air. So absence of oxygen, so no free oxygen is required by them, and anaerobic bacteria are killed if exposed to air. Finally is the reproduction part. Reproduction in bacteria is only asexual by means of fission or cell division. So the reproduction in case of bacteria will only be by cell division. So there is one bacteria which breaks down into two, then this two break down into 4, and then 4 dividing down into 8, and then 16, and then 32. So that is binary fission which is going to take place in case of cell, in the cells of the bacteria. So cell division is the only method. So it's by asexual method of fission or cell division. The circular DNA duplicates. The cell expands and the two DNA are pulled apart. The cell constricts at the middle, separating the two cells. Depending on the type of bacteria, the two daughter cells may either remain attached or may separate from each other as independent cells. So depending upon the cell, the type of bacteria, they will either, like अगर समझो वो डिप्लोकोकाई वगैरह रहेगा तो वो साथ में रहेंगे, पर अगर ऐसा नहीं है तो what will happen, they are going to means if they are not going to be in clusters or something, they are going to remain individual. So so one cell is there, this one cell is going to elongate and the DNA material inside is going to duplicate. So एक के बदले 2 हो जाएगा, and then it is going to separate out, and then there will be a furrow which will be formed over here, and it divides into 2 separate bacteria. So in this way the bacteria are going to be reproduction taking place asexually in case of bacteria. And this reproduction is very, very, very fast. You can just imagine this thing, that if you, your mother, when she prepares the curd, so she just takes one complete container of milk and just adds maximum or 1 tablespoon of curd inside it and stirs it and keeps it overnight, and the next morning the whole of the milk turns into curd. So you can imagine that 1 tablespoon got converted into 1 complete vessel or filled with curd. So that's why we can see that the bacterial growth is enormous. The enormous growth is there in bacteria. Under ideal conditions, some bacteria divide at a very fast rate, once in every half an hour. At this rate, of growth and division, starting from with a single bacterium at the end of 24 hours, there would be, now that number is something which we cannot even find out that kitna billion and kitna trillion वगैरह है, but it's a very big number of bacteria. The next is highly primitive type of sexual reproduction. So there are certain kind of bacteria which do show certain kind of sexual reproduction. So in case of sexual reproduction, so a highly primitive type of sexual reproduction has been found in very few bacteria and is extremely simple. In this method, two bacteria of different strains but of the same species come together, that is conjugation. The plasmids, a small circular DNA molecule different from the chromosomal DNA of the donor, may be transferred into the recipient through a hollow tube-like extension of the donor cell. So there will be two cells over here, और यहाँ से plasmid में से एक particular kind of a strain of the DNA is going to come and enter into the DNA of the other one, and hence there will be a sexual reproduction taking place between them. So there will be a small tube-like structure between the two bacteria, and hence the donor say DNA say it will go to the recipient DNA, and formation of the new cell is going to be formed. So this is how the highly primitive kind of sexual reproduction takes place in certain kind of bacteria. Now another very important thing what happens in bacteria is the formation of spores. Now spore formation in case of bacteria is not a method of reproduction, but it is only a method to protect itself from unfavorable conditions. Bacteria, they have got a certain bacteria, they have got the capacity that they can be in extreme climatic conditions, and some of them can even be present in case of acids. Okay, they can even withstand acids or high alkalines. So in such cases, what happens is the bacteria which are there, they will be encapsulating themselves in a particular kind of like a cocoon sort of thing, which is going to be called as a spore. So when the unfavorable conditions are available to the bacteria hides itself in that particular kind of spore, and when the conditions are now again suitable, it comes out of the spore, thus saving itself from that particular extreme kind of condition. So this is how the bacteria is going to protect itself, but this spore formation is not a part of reproduction. It is just overcomes the unfavorable conditions. Spores to overcome unfavorable conditions. When unfavorable conditions set in, such as drying up of vegetation, the bacterial cell draws its content into a spherical mass which becomes surrounded by a thick and hard protective wall. Okay, so there will be a, whenever that condition is available, there at that time the cell is going to, the bacteria will be covering itself by a thick protective hard kind of a covering over the cell wall. This little rounded body is a spore which is contained within the original cell wall, but later becomes free when the cell wall ruptures. Such spores are usually the resting stage which can withstand unfavorable conditions. These can tolerate extreme dryness. Some cannot be killed even at a temperature of boiling water or frozen ice. Some can tolerate poisonous chemicals. The spores can be carried far and wide by various means such as wind, water and contact. On meeting favorable conditions, the spores germinate and then dormant cytoplasm becomes active and streams out of the ruptured wall into the form of new bacteria. So this spore is such that it will be just be able to travel anywhere throughout the area without any problems. They can go through wind with the help of the wind or maybe water anyways, and once the favorable conditions are available to it, it can break open and rupture and it can form into a new bacterium. So spore formation in bacteria is not a method of reproduction but simply a method of escaping unfavorable conditions. So that is about the bacterial part. So now we will see the use of bacteria in medicine. So that is the next part, that is use of bacteria in medicine. But in the next video